Okay, we are now protein synthesis video 3. So we were talking about the chaperone proteins carrying the messenger RNA to the small subunit of the ribosome. That's where we were. All right. The small subunit of the ribosome then slides underneath the messenger RNA. Let's see what we can find here. The small subunit of the ribosome. Let's find a picture here. Let's find a picture. Is going to slide underneath, say right here. This is the RNA polymerase right there, but I'm going to go further here. Okay, let's go further. Okay, so here's a picture here you can look at. Right here is the small subunit of the ribosome. The initial messenger RNA, you go through attaches to that. Then the small subunit of the ribosome starts to slide underneath, and each of these prongs are nitrogenous nucleotides, nitrogenous bases. When it slides underneath, it goes through the reading frames. The first three are going to start at what we call the start codon. In other words, when the message comes out, now let's think about it, it's a long Western Union message. Remember, it had the capping units. So when does the reading actually start to say it's time to start constructing the polypeptide of protein, because you got some other units on the ends. That's what we call the start codon. As the small subunit goes underneath, it gets to something we call the start codon. The start codon. Now let's go back to our codons real quick. Let's go back to our codons. Here are our codons. And AUG can code for methionine, but it's also the start codon. It can code, so that means on the beginning of every polypeptide of protein, you put methionine because it's the start codon. Does everyone need it on the beginning? No, so if it's not part, it'll cut it off later, but that is our official start codon. So that's how the messenger RNA knows where to begin. Then it will start sliding underneath. Now the transfer RNA brings in methionine, and then it slides further. When it slides further, it goes to the next three. And then another transfer RNA comes in, and brings that particular amino acid in. Whichever is the next one in line. Whatever's the next one in line. And then it just keeps going and keeps going and keeps going. See, here would be where you're bringing a new one in, but each time you bring one in, now by the time that you start, once you get to the second one, this was the beginning, then the large subunit comes in. When the large subunit comes in, the large subunit has enzymes within it that puts the peptide bond between the first amino acid and the next one. And then it goes further, and each time you come in, you read a new one, bring one in, that's the growing tail right there. Here's the transfer RNA that brought something in. Once it hooks to the end, it leaves. And then you start. So essentially what you're doing is bringing, hooking, and then the one that brought it in before. So I need a new one coming in. Next amino acid to be coming in. Here's the one exiting. So here would be the deal. I'm coming this way, 3 to 5. Let me start it this way. I'm coming 3 to 5. I'm bringing in my new one. That's the new amino acid. That's the A. Then it's going to move, and then that would hook to the growing amino acid chain. Then that would move here to the exit one, and he's gone. So bring new, hook to what you got, and the one that brought the one previous exits. The one that brought the one previous exits. Now, you don't have to get real heavy into this. Just kind of get an idea of flavor. It's kind of hard to do it on your own, I know. But what you're attempting to do, let's go back, is to make a polypeptide or a protein. The key players are the DNA. It has the code. The DNA, let's go back to a drawing. That kind of puts it all together. So we start first, you have your DNA. Then you look for where the gene is located, on which chromosome and what gene loci within that chromosome. Then the job, an RNA polymerase enzyme does transcription. It transcribes the DNA to a pre-messenger RNA. Why they call it pre? Because you still have those introns. Once you cut the introns out using the spliceosome, it becomes a good messenger RNA. At that time, it then exits through the nuclear pores and is picked up by a chaperone. The chaperone then helps take it to the small subunit. The large subunit has not attached. When it takes it there, then that's when you start the construction, which involves using the messenger RNA as the now copied blueprint, the construction table as the ribosome, and the transfer RNA is bringing stuff in. Now, translation. When you went from nucleotide talk on the messenger RNA to bringing in transfer RNA to bring in the amino acids, you're speaking from one language to another language. You're speaking from nucleotide talk into amino acid talk, from nucleotide talk into amino acid talk. Okay? So the whole process keeps repeating itself. So if you read through, I tried to put this in a sequential order. What you want to do is go through and just kind of read through. Don't get real heavy. I'm not going to test you a lot on no protein synthesis, but I would like you to understand it to some extent. I would like for you to understand it to some extent. Now, once you go so far, so let me go down here. So it comes in, keeps coming in, and then there'll come a point in time, I can find it. So here's the process again. I have many pictures of it. But it'll come a point in time, go further, that the whole polypeptide will exit. So termination of translation, right here. And then the polypeptide is freed. So you have a termination signal that says, hey, don't put another amino acid, let it go. Now, I want you to understand that when you're doing this process, it has to happen real quickly, because out in the cytoplasm are many hostile features. So you don't do this and just create one. What happens is you have what we call polysomes. That means on the messenger RNA, a ribosome will attach and start doing its thing. Then another one will attach right behind it in tandem and do its thing. They're all making the same polypeptide, but you're trying to make a bunch of them at one time. You don't do protein synthesis to make one. You see all these, you have to be down homeostatically, maybe 1 million. So when you do it, you're doing it quickly. And that's what we call a polysome. It's a bunch of ribosomes lined up doing the same thing to make a bunch of them very quickly, because this message out here is being torn up fairly quickly in the cytoplasm. So when we look at this here, and I encourage you to go through, you can kind of get an idea of flavoring of how this protein synthesis works. Now again, don't get real heavy into it. So don't get all bent out of shape and think, oh my God, it's not like that. The termination occurs when a stop codon in the met reaches the A site of the ribosome, and basically it stops and just puts an enzyme to let the message go. Okay. Now we'll go a little bit further. All right. So remember what I said. If a protein or polypeptide is made on a floating ribosome, it's used inside the cell. That means that when it terminates, it just lets it go into the cytoplasm to be used for whatever there. But here's what happens if it's going to be on a fixed. If it's made on a fixed ribosome, it's to be used in the cell membrane or outside the cell. Okay. So what happens is a fixed ribosome doesn't know it's fixed. There's what we call a signal sequence, which means, listen, you can read on this. When the message first starts being made, considering there are receptors on the rough ER, not on the smooth, then once you make a few amino acids on the poly, not the whole thing, just a few, that pulls the whole message to the rough ER. So it's the protein being made that pulls the whole thing over. It's not the ribosome. And when it pulls it over, it pulls everything, including the ribosome. So some ribosomes attach to the rough as the poly being made. The developing pulls the messenger RNA and ribosome to the ER in the region that is to be rough. It is the first amino acids of the developing polypeptide that pulls it. That means we've checked it out. If we cut those first few amino acids, it won't attach, and it'll let it off in the cytoplasm. So all ribosomes are the same. There's no ribosome dedicated to be fixed. The polypeptide being produced determines whether the ribosome will do it. And that's what this is showing here in this set of drawings, is how it's pulled over. And remember, anything that goes through the rough ER will go through the Golgi, because what the Golgi is going to do is take it to the cell membrane or make a vesicle to exit it outside the cell. Okay? So I won't get into that there. All right, so go through, just kind of go through, and in a loose way, but read your text. Don't get real heavy, but go through and just kind of get a fairly decent but not exact understanding of protein synthesis. We do most of this in general bio, so for those that haven't taken general bio, I know it's very hard to do. So just try to kind of get an idea of how it happens. But remember these features. DNA, the only job DNA has is to code for the primary structure of a protein, or polypeptide. The primary structure is the linear sequence of amino acids. The linear sequence of amino acids determine what the secondary, tertiary, and quaternary, because it will determine how it's going to bond to get to the final folding. In protein synthesis, the first thing you have to do is find where in the heck the gene is. Which chromosome is it on? And in the chromosome, where's the file drawer, which is called a gene loci. Then an RNA polymerase enzyme will read that. The RNA polymerase then will read DNA into RNA, which we'll call transcription, one language to the other. Then what will happen is you'll make a pre-messenger RNA. Why? Because you got the introns in there. It does not need to leave the nucleus because it's an ineffective protein if it's made from that. So then enzymes come in, cut the introns out, called a spliceosome, and hook the exons. The message now is a complete messenger RNA can exit the nucleus through the nuclear pores. It goes into the cytoplasm, and chaperones carry it to the small subunit of the ribosome. The small subunit of the ribosome starts working on it as a construction table, and finally the large subunit comes in. The job of the large subunit is to have enzymes to place the peptide bond and some other things. Then it'll start the message at the start codon, AUG, move on down till it gets to the termination. But several ribosomes, because you need to make it quick and furious, will hook on, and that's what we call a polysome. So kind of read on that if you would.